So welcome. Bienvenidos a nuestro simposio hoy. So I was introducing Dr. Gusev, who is statistical geneticist and associate professor of medicine here, Dana-Farber, and also in the population sciences division. He received his PhD training in computer science from Columbia, followed by a postdoctoral fellowship focused on developing computational methods to understand disease heritability at the Harvard School of Public Health. His lab has led the development of methods for deciphering biological mechanisms of cancer risk from genome-wide association studies, identifying germline somatic interactions, and characterizing the germline influence on response to therapy. His recent work includes the identification of biomarkers of immunotherapy toxicity, and this is something that I'm a big fan of, is his development of predictive algorithms for cancers of unknown primaries. A lot of people in this room don't know, if you haven't taken care of patients, that we have some patients who show up with advanced cancer, and everything in oncology is based on where we think a cancer came from in terms of what agents to use. So this is a population, it's a small population that we really don't know what to do. We've developed algorithms for chemotherapy based on what we think it's most likely. We test, sometimes the cancers are very confused, and so they don't even know what tissue they came from in terms of none of the tests work. And so it's kind of a shot in the dark. So the work that Sasha is doing to try and figure out where these cancers are most likely from could not have more impact for that group of patients. So I really think that's very important work. And anyway, so I'm a fan of that, but all his other stuff, some of which I don't understand, but we're going to actually get some lecture today about it. So he works on that. He also works on integration of genetic ancestry to improve existing biomarkers. So he's going to talk to us today about characterizing germline cancer mechanisms at scale, risk, tumor evolution, and treatment outcomes. So welcome, Dr. Gusev.
Thank you, Goya. Thank you guys for having me. I'm super excited to present on this work. And actually, I won't be talking about the cancer of unknown primary research, but we are continuing to work in that area, including bringing in data from digital pathology and other modalities. So I'm very happy to chat about that after the talk as well. It's something we're really excited about. Today, I'm going to be talking primarily actually about that final point, which is the integration of an understanding of genetic ancestry in the study of tumors, both from the perspective of risk, tumor evolution, and clinical outcomes, and how we can incorporate genetic ancestry into how we study and understand disparities. And so in particular, great. Okay, so I'm going to try to get through several different vignettes. Hopefully, I can do this in a way that's efficient but not confusing, that highlight different ways of thinking about genetic ancestry in this context. And so first of all, I'm just going to introduce the concepts of genetic ancestry and the distinction between race, ancestry, and genetic causes, which I think is important to sort of keep in the back of your mind through the entirety of the talk. And then I'm going to talk about some work that we've been doing, trying to understand the correlation between genetic ancestry and tumor evolution. How do cancers evolve? How are they formed? As well as the correlation between genetic ancestry and clinical outcomes, and how we could be incorporating ancestry and genetic diversity into our models, predictive models. And then finally, I'll talk about an example of an existing biomarker. This is a bit more of a technical example, but where factors related to genetic ancestry actually have led to biases and false positives in the way that we currently use biomarkers in clinical prediction, and how thinking about ancestry, even in a purely somatic setting, can improve and make the tools that we use more generalizable. So this is hopefully kind of highlights different ways that we can use ancestry to make interesting discoveries, and then also refine and broaden the use of the tools that we already have. Okay, so with that in mind, I think the first point, I've been using the term genetic ancestry a lot, but it's actually important to sort of get the definitions and the concepts down first. So an important caveat that I want to make is that there are distinct concepts of race ethnicity, which is a social construct, genetic ancestry, which is a relative genetic measurement, and genetic causes, which are often correlated but not necessarily a function of genetic ancestry. And specifically, the way that I think about these ideas, I'll kind of, I usually think about directed acyclical graphs or DAGs, so I'll kind of outline in that same form here, is if we think about race as a social construct that exists within a specific social context, when we think about disparities, we're often interested in understanding how race is correlated or factors related to, for example, social determinants of health or racial discrimination lead to clinical outcomes. Separately, we have this concept of genetic variation, a biological variable that exists in all humans. And there may be biological factors driven by genetics that also correlate with clinical outcomes. And then we have a measurement called genetic ancestry, which is in fact essentially a measure of how similar a given target individual is in their genetic variation to some reference population. And this idea that genetic ancestry is actually essentially a relative quantity, I think kind of gets lost in these discussions a lot. But anytime we're analyzing ancestry, we first have to define some set of reference groups, and then we ask, are you more related to this reference group versus this other reference group? And then as a consequence of the way that race as a social construct has been used in our society, there are of course going to be correlations between racial groups and labels, which change over time, but will be correlated with genetic ancestry. And so all of the arrows in this diagram themselves start to have fairly complex relationships. And it's sort of important to keep in mind that when we see correlations, that does not necessarily mean that a factor that's correlated is actually causal for the outcome. And I'll kind of get into how we try to disentangle correlation from causality in this setting. So with that in mind, how do we actually get this kind of data? I think a key bottleneck to studying and understanding genetic ancestry, in addition to the race ethnicity measures that we get from clinical data, has been that you actually need genetic variation to estimate these kinds of measurements. And a lot of the data that has been collected, especially here at Dana-Farber, has been tumor-only sequencing data, typically from targeted panels. And this is data that's not usually thought of as containing enough information to capture components of genetic ancestry. And so some of the most foundational work that we've been doing in my lab is just showing that you can, in fact, infer genetic ancestry and use it reliably from data that people typically do not think of as containing that information. Specifically, here at the Farber, there's been a long-standing effort to sequence patients, typically with advanced disease, on a targeted panel called OncoPanel, which is measuring somatic variation in several hundred genes. That effort is now up to close to 40,000 patients that have been sequenced. All of them are linked to their electronic health records. And so this potentially provides a very useful resource to study relationships between genomic variation and clinical outcomes. Additionally, we have shown that you can actually take these tumors, which are sequenced on a targeted panel, and then integrate them together with a germline reference panel to infer the germline genetic variation that these patients have. And I kind of won't go through the whole statistical process that we are using here, but the basic idea is that these panels measure a lot of sequencing material from the targeted genes, from the couple hundred important cancer driver genes, but actually a lot of the sequencing material lands off target in the rest of the genome. Generally, the typical workflow is to just throw away that off-target content. But it turns out that if we integrate in haplotype reference data from existing public databases, we can do inference on these low coverage sequencing reads in the rest of the genome. And we can use this variation to infer actually surprisingly accurate estimates of germline polymorphisms that are not in the targeted regions. And now that we have this kind of probabilistic estimate of inherited germline variation for these 40,000 sequenced tumors, we can then integrate an additional ancestry reference panel. As I mentioned, genetic ancestry is always defined relative to some population and infer sort of broad continental components of ancestry in these individuals. And so here I'm showing that imputed data being used to infer continental Asian ancestry with a Japanese reference panel and continental African ancestry with a Yoruban reference panel. And the inference that we get from this incidental tumor imputation, it basically is, perfectly consistent with the inference that you would get from an actual germline SNP array, which is the ground truth for measuring this variable. So this kind of quantity is something that we can get with very high accuracy from this off-target data. And again, I'll just kind of pitch this as something that may exist in data sets that you have already collected and that are working with. Certainly exome sequencing can be employed in this way. Other targeted panels, RNA sequencing, even epigenomic sequencing. If there is off-target coverage, you can oftentimes infer genetic ancestry for those individuals. Additionally, once we actually start looking at the ancestry components that we infer, we see that there's quite a lot of diversity even within this sort of single institution population. And so there's a couple of different ways that we can visualize ancestry. One way that's commonly used is these principal component visualizations, which try to extract the sort of major drivers of genetic variation in these individuals. And you can see here each point is a person. They're color-coded by their recorded race in the electronic health record. And there are these sort of clines of African and Asian ancestry. And there are deeper clines if you go to further dimensions that are harder to visualize here. And then kind of coming back to the point that race and ancestry are correlated but distinct factors, when we break these individuals down by their recorded race in the health record, we see that individuals who are recorded as Asian actually have a sort of cline of inferred Asian ancestry, including some components of African ancestry and European ancestry. Individuals who are recorded as Black have differing and varying proportions of admixture of African and European ancestry. And even some subset of individuals who are recorded as white actually have a predominance of non-European ancestry or again, admixture for multiple reference populations. And so in this case, the genetic ancestry gives us a granularity of measurement that is distinct from the kind of labels that we have from clinical data. Okay, so that's kind of the foundation for these analyses. And then the first example that I want to talk about, some work that we've been doing in understanding the relationship between genetic ancestry and properties of the tumor. Somatic components of tumors and then eventually clinical outcomes. So in general, it's well known that cancer subtypes differ across different populations. And a question that we wanted to ask is whether this goes beyond the level of subtypes and into specific associations between ancestry and somatic features. And also whether there are relationships between ancestry, somatic features, and clinical outcomes. The data that we'll be using is the sort of cohort that I already talked about, this profile sequencing effort, which at the time that we did this study included about 30,000 patients. The ancestry components I've already described. And sort of just to cut to the chase, we do, in fact, and this has been observed in the literature, see significant associations between genetic ancestry and specific driver mutations in these tumors. So for example, on the left here, and sorry, this is a little bit blurry, but I'm showing an association between East Asian ancestry and EGFR somatic driver mutations in patients with non-small cell lung cancer. And you can see that individuals who are most similar to a European reference population have an EGFR mutation rate of about 15%, whereas individuals who are most similar across their entire genome to an East Asian reference population have an EGFR driver mutation rate of about 75%. So there's actually a really substantial difference in the type of tumor and the type of driver that you develop as a consequence of these ancestry components, or really, again, I should say, as a correlation of these ancestry components, because the precise factors that cause these mutations, we actually don't know. And we see similar examples for other driver mutations. Again, this is here, we're just replicating findings that were previously observed in the literature, but this KEAP1 driver, we see at a lower frequency, at a much lower frequency in individuals with East Asian ancestry than with European ancestry. And when we expand this type of analysis to 5 common cancer types across 2 different cohorts, so here we were using data from Dana-Farber, as well as a collaboration with Tempus with a similar kind of imputation, we actually see a large number of somatic associations. Each of these points that has a little label next to it is a FDR, false discovery rate significant association between a somatic driver and ancestry proportions. We see in pretty much every cancer type, there are some somatic drivers with differences as a function of ancestry. So this phenomenon where you have molecular differences in tumors in different populations seems to be quite widespread. Okay, so the second question that we wanted to ask is, to what extent does this relationship actually influence clinical outcomes and meaningfully affects what happens to patients? And to kind of set the scene for this, there's 2 ways that you could imagine a somatic driver influencing clinical outcomes in these diverse populations. One is, as we've seen, East Asian ancestry is associated with EGFR driver status. It could be the case that EGFR drivers have sort of the same kind of survival curve in, regardless of which population context you are in, but because East Asian individuals have more of these drivers, they have survival curves that look more like this one over here. So this is effectively a driver that has a consistent effect on survival, but that has a higher frequency in one population than another. An alternative model is one where there is actually an interaction between ancestry and the driver status. So individuals with European ancestry who have an EGFR mutation tend to have this kind of survival curve, whereas individuals with non-European ancestry who have an EGFR mutation tend to have this kind of survival curve. And again, the mechanisms of this could be environmental factors, for example, that are different between these 2 individuals, or other genetic interactions with ancestry. And environmental factors really can, there's a whole sort of can of worms of what possible factors those can be, including when these patients get diagnosed, how much lead time bias there is until they're sequenced, which kind of treatments they receive, how much access they have to the healthcare system. So there's a lot that I'm putting into this bucket of environment, but this kind of interaction would be indicative of essentially some kind of environmental modifier to this driver status. So we wanted to ask quantitatively whether there's evidence in the data of this second model. And the way that we did that is by employing a survival analysis, incorporating all of the features that I just described. So a kind of conventional biomarker association analysis looks at survival, for example, overall survival as the endpoint for a population of individuals, and then asks if there's an effect or an association of a somatic mutation with this overall survival, with the time until mortality. So we're asking if this mutation increases or decreases your propensity, the survival propensity. The model that we're employing now incorporates covariate for genetic ancestry, so this continuous factor relative to a reference population, and then an ancestry and mutation interaction term. So this is the sort of what I was trying to illustrate in the toy example previously, is we're asking if your global genetic ancestry actually may be modifying the influence on survival of the somatic mutation that you have. And when we do this, the first thing that we observe is when we just plug this model, we plug these features into a model, again, in the clinical data, largely from patients treated at Dana-Farber, without somatic mutations, we see that there is already a weak association between genetic ancestry and survival. So specifically, African ancestry is associated with poorer survival, and this is largely driven by the significant effect in breast cancer. I think this has again been observed in the literature, possibly explained by differences in breast subtypes in these individuals. And then East Asian ancestry is associated with significantly better survival. Again, these are sort of only nominally significant associations, and that is driven largely by an effect in lung cancer. So just in the baseline population, we are already seeing some, although modest, survival differences as a function of genetic ancestry. Now, when we plug in this interaction term and we basically scan across every driver mutation and ask if there are significant interactions with ancestry, indeed, we do find these kinds of interactions. And I'm going to show a couple of examples here with Kaplan-Meier plots. So if you look at the figure on the right, this is looking at patients with glioma who are wild type for this PRDM1 copy number alteration. And you can see that actually in this cohort, individuals with a predominantly African genetic ancestry have better survival than individuals with European ancestry. On the other hand, when we look at just the subpopulation that has a PRDM1 somatic copy number alteration, these curves are basically reversed, and individuals with African ancestry have worse outcomes than individuals with European ancestry. And so this highlights the phenomenon that ancestry can modify and change the influence of these somatic alterations. And I'll show another example where we see the opposite. Now we're looking in ovarian cancer, and in this case, individuals with this RHEB1 somatic copy number have similar survival curves as a function of ancestry, but the copy number alteration produces a significantly different survival curve as a function of ancestry. So again, this driver appears to behave or respond differently to treatment in different populations. And when we kind of broaden this out and do a scan across all of the cancer types, the 5 cancer types that I described, we see that this phenomenon is, again, quite pervasive. There are many, each of these points is a different somatic feature that we tested. There are many somatic features that tend to have an interaction with genetic ancestry in how they influence clinical outcomes. So with that in mind, that kind of massive volcano plot is pretty hard to interpret. One question that we wanted to ask is if we put all of these interactions together into a single model, how much of an influence do they have or an association do they have with clinical outcomes in total? And so the way that we did that is now essentially expanding that interaction model that I described first to contain all relatively common somatic drivers in this population. So we have now a big matrix of mutation status. All of the drivers that are present here are encoded. And then we have a big interaction matrix. We're looking for all interactions between genetic ancestry and mutation status. And under the hood, we're running a machine learning model that sort of appropriately penalizes and fits all of these parameters together. When we fit this model without the ancestry component, what we see is that, and again, this is something that's been observed in the literature, it is possible to predict patient survival using these somatic features alone. So across the cancer types in a sort of meta-analysis, we see that a genetic predictor built out of all somatic mutations in this penalized way yields a hazard ratio of about 1.7. That's highly statistically significant. And that at least seems to kind of trend in the same direction across all of the individual cancers as well. So this is just demonstrating the basic point that the somatic features that you have in your tumor do correlate and are predictive of your survival outcomes. But when we bring in these ancestry interactions, we again see a significant a statistically significant effect of this interaction component. And we see that the hazard ratio is actually pretty similar to the one that is observed for the marginal model, a hazard ratio of about 1.5 for the interaction between all somatic features and genetic ancestry in this population. And so the number of non-European, the sort of proportion of non-European patients in this cohort is quite small, which is why the statistical significance here is a lot weaker. But on a sort of per dose effect, we see that incorporating these ancestry interactions can significantly influence the way that we sort of predict patient level survival. And again, to kind of make that maybe even more clear, we plotted the change in predicted survival for patients between a model that includes only genomic information and a model that includes these ancestry somatic interactions. So if we sort of improve the model to incorporate ancestry, how much of a change do we get on average? And we, kind of looking at this distribution across different cancer types. And the point really here is that depending on the cancer type, between 10 and 30% of patients have a change in their predicted survival that's greater than 6 months. So these values here are in months. So there's a sizable proportion of the population where a model that just uses genomics is off by 6 months relative to a model that incorporates genetic ancestry interactions on top of genomics. And so again, I just want to make the point that what we're seeing are associations with ancestry. They do not necessarily need to be actual genetic causes. As I mentioned, ancestry is correlated with a lot of other environmental factors, and a likely interpretation here is that it's actually those environmental factors that modify the influence of these somatic drivers. But the point is that this is sort of a missing piece in our predictive models that can have a pretty substantial effect. And so just to conclude this example, I hope that I've kind of given you some demonstration that, first of all, genetic ancestry is interesting to study and can be inferred from the kind of data that we routinely collect, that there is an association between ancestry and the type of tumor and the drivers that patients develop. And additionally, there is this interaction between ancestry and somatic features in terms of their effect on clinical outcomes. And this interaction may actually can be leveraged to improve the performance of these predictive models. So that was an example that was kind of, that was primarily focused around discovering associations and trying to identify potential novel mechanisms of how ancestry may be influencing clinical outcomes. The next example that I want to present is one in which ancestry actually introduces biases in existing clinically approved biomarkers, and how incorporating ancestry can help address those biases. And specifically, I'm going to be talking about tumor mutational burden, or TMB, in the context of the treatment of immunotherapy or immune checkpoint inhibitors. And the questions that we wanted to ask with this study are, first of all, often in many cases, and in particular, this was the longstanding case at Dana-Farber, tumor mutational burden, which is basically the total number of somatic mutations that a tumor has, is measured using sequencing only of somatic or tumor data. We wanted to ask how accurate these estimates were across different populations, and whether the differences in accuracy have an influence in how predictive this biomarker is. And this is an important question, or I think it's an important question, because TMB, or specifically TMB high, patients with tumors with patients with tumors greater than 10 somatic mutations, is one of the few FDA-approved biomarkers, and has been now for several years, for immune checkpoint inhibitors across a wide range of solid tumors. So this is kind of this big pan-cancer biomarker for an important treatment, and so it's important to understand whether it is in fact accurate or not. And I'm not going to kind of talk through the immunology, but just to give you a very, very simplistic idea of why of the model for this biomarker, the thinking is that a higher mutational burden or higher TMB leads to more neoantigens presenting to the surface of the tumor, and so produces a tumor that is easier for the immune system to identify. Immunotherapy then activates the immune system, and so it's able to find those tumors and neutralize them. Whereas if you're TMB low, you have few somatic mutations, it's hard for the immune system, for the T cells to identify those mutations. So the immunotherapy doesn't work as well. This is the basic idea. And I think the key takeaway here is that what we really care about is accurate estimates of this sort of neoantigen somatic presentation in the tumor. We don't care about other mutational factors that don't relate to the tumor. This is sort of foreshadowing to the biases that I'll be talking about. And so to kind of understand where ancestry fits in here, it's important to first understand how we actually do somatic variant calling and TMB estimation in these clinical sequences. The basic idea is that when we look at a sort of tissue biopsy, what we're actually observing is a kind of clump, a collection of cells that is a mixture of malignant cells, these tumor cells in orange here, which contain somatic mutations, and then also just healthy cells from the sort of stroma and microenvironment and the blood and the other tissue around the tumor, which do not contain the somatic mutation. And then additionally, in some cases, we're looking at a variant that is actually a germline polymorphism that's just present in individuals because they inherited it from their parents. And that germline mutation is going to now be present in all of the cells that are sequenced as part of the biopsy. And these are the mutations that we care about. These tell us about this neoantigen presentation. And these are the mutations that we don't want to identify because they're telling us something about host factors that don't contribute to TMB. And so an interesting observation that we made is when looking at different types of sequencing platforms, you actually can observe a different mix of the inferred somatic versus these sort of false germline variants. And the way that we do that is by looking at the distribution of the variant allele fraction. So the number of, for a given called somatic mutation, the number of reads that that support the somatic mutation as a function of the total number of reads. So for a true somatic variant, we expect this distribution to be below 0.5 and usually quite a lot below 0.5 because tumors are impure. Most of the time they contain a lot of other cells. And so when we look at data from Sloan Kettering, which is a gold standard tumor normal matched filtering process, we see exactly that distribution. Most of the mutations that get called have a median variant allele fraction of about 0.25 because it's, they're in half of the tumor cells, and then the tumor cells are only about half of the tissue that was collected. And we also see no differences here, or no significant differences as a function of the patient race in this cohort. We didn't have ancestry available because this is public data. On the other hand, when we look at data that was collected at Dana-Farber from tumor-only sequencing, where you didn't have this separate germline filtration step, what we see is a very different distribution, and specifically this kind of bump at a variant allele fraction of 0.5. And this bump essentially reflects a mixture of false positive germline mutations. Germline mutations are going to be present in all cells. We expect them to be at a variant allele fraction of about 50%. And so what we're seeing here is basically a mix of this sort of ground truth distribution and then a bunch of germline false positives that are showing up. And again, this is sort of fully called and processed data that we just downloaded from the AACR Genie website. This was intended to be high quality somatic variation from a variety of cohorts, but obviously includes this, this sort of confounding. And then the additional important observation here is that when you compare across the distributions across these self-reported racial groups, you see that non-white patients actually have big, sort of a higher variant allele fraction and a larger mix of these germline false positives. And I'll explain why that is, but I think it's just important to note that this bias of germline miscalling, not only is it present, but it is actually present in a different fraction depending on which population you come from. And just to sort of convey that this is not simply a function of something that's happening in the Dana-Farber sequencing data, when we looked across all of the other tumor-only sequencing cohorts, we see this same phenomenon. There's basically a bump of germline false positives. And that bump can differ depending on the sequencing platform, and it also tends to differ across the populations. So now coming back to why do we see this bias as a function of self-reported race? In order to kind of understand that, we need to step back a little bit as to how these variants are actually being called. So when we do the sort of gold standard tumor normal sequencing, the variant calling algorithms identify all of the mutations in the tumor and then subtract out anything that they observe in the corresponding normal sample. When we do tumor-only sequencing, we identify all of the mutations in the tumor, and then we don't have a normal to compare to, and so we actually subtract out mutations that are observed in an external reference panel. This again gets back to this sort of observation that a lot of times what we're doing is based on reference panels and not the observed individuals. And as a sort of, this is true for a lot of cohorts and data sets that have been collected. These reference panels severely underrepresent the contribution of non-European ancestry individuals, and specifically the panel that gets used here, which is from the EGSAC consortium, has 6 times more samples from European ancestry than non-European ancestry. And so what happens is if you're thinking about these tumors as coming from patients with a diverse ancestry proportion, there's some individuals who are predominantly European ancestry here, there's some individuals who are admixed, and there's some individuals who are predominantly non-European ancestry. When we do this tumor-only filtering, what we want to do is find these green somatic driver mutations, these green triangles. And we want to exclude out these black germline variant dots. When we use a reference panel, so first of all, sorry, when we first call, we can't distinguish these. We just see all of the mutations. Now, when we bring in a germline reference panel, which is overrepresented for one population, what we see is that this works quite well for the European ancestry individual. It's able to spot that this mutation and this mutation are present in the reference panel, exclude them, and give them an accurate estimate of their TMB. When we now incorporate, evaluate individuals with non-European ancestry, it works reasonably well. It's able to spot this common germline variant that they see. But in instances where they have germline variants that are not that are on a ancestry segment that is underrepresented in the panel, that variant gets miscalled. And so they get an inflated estimate of their tumor mutational burden. And then finally, when we look at an individual who is predominantly non-European, we see even more of these false positives because their germline mutations are underrepresented in this reference panel. And so this is actually kind of an interesting phenomenon. We're talking about tumor mutations, but we get these biases as a function of germline reference data. And then additionally, you could imagine, I don't know if I have this here, you could imagine maybe correcting for this problem by downsampling the European population here or collecting more data from non-European reference individuals. That's definitely a good thing to do, but actually you would need to collect even more data from non-European populations because of population genetics history. There's more diversity in African genomes than there is in European genomes. The average African ancestry individual has more germline variants. And so this is really kind of flipped as to what we actually need, which is more reference panels from non-European ancestry individuals. So that is just to kind of convey the point of we observe these biases in real clinical data that is intended for biomarker analyses. And we now kind of understand that these biases are a function of ancestry and different underrepresentation in the reference panels that are being used. The final question that we wanted to ask is whether we could actually try to correct for these biases using the existing data. And the way that we did that is we went to the TCGA, the Cancer Genome Atlas, where we had data from tumor and normal match sequencing from a multi-ancestry population that we could then use to sort of train a recalibration score. So we looked at TCGA data, we computed the true accurate tumor mutational burden estimates from these individuals, and then we hid the match normal data and recomputed what you would get with a tumor-only estimate of the tumor mutational burden. This is inflated, and then we learn the relationship between the proportion of ancestry that an individual has in total and the level of inflation that they had. And then the idea is now that when we have a new sample that has only a tumor-only TMB estimate and the genetic ancestry inferred in the sort of way that I described at the beginning of the talk, we can now apply this recalibration and get a corrected estimate of their TMB. Now, again, ideally, we would actually have match normal sequencing on this individual. We're making a probabilistic correction, but this gives us an idea of how much their estimate could be inflated as a function of ancestry. So we applied this technique again to data from Dana-Farber to get a sense of how much of an impact does this actually make in real samples. Specifically, because immunotherapy is still approved, or at the time was approved for only a subset of tumors, we focused on cancer types that do routinely get immune checkpoint inhibitors. We looked at data from about 2,900 sequenced patients, and we estimated their, we looked from a larger population of which about 2,900 were TMB high, so would have qualified for immunotherapy. And then we applied this TCGA-derived correction to their raw tumor-only TMB estimate. And we evaluated how many initially were thought to be TMB high, and then were recalibrated to TMB low. And when we do that in the individuals with European ancestry, we see that about a third of them are actually being miscalled as TMB high as a function of these germline false positives, which are present in every individual, just as a function of the reference panel. But when we expand this to individuals of other ancestries, we see that individuals with predominantly Asian ancestry have about a 40% chance of being miscalled as TMB high, and individuals with African ancestry have close to a 50% chance of being miscalled as TMB high. So there are biases in this estimate, and those biases differ depending on the population. And the reason is because of this sort of underrepresentation in the reference panels that are being used. And then the final question that we had is, in addition to these biases in the biomarker itself, do they actually have a consequence for clinical outcomes for these patients? Maybe the estimate is miscalibrated, but it doesn't really end up mattering ultimately in terms of survival. And so the first thing that we did is just, again, confirm the established association in the literature. The reason why this is a biomarker is that individuals who have true or sort of corrected high tumor mutational burden do indeed have longer survival on immunotherapy. For this, we went to two different cohorts, one at Dana-Farber, one at Sloan Kettering, and we see in both TMB high is associated with longer survival. This is great. This is the sort of expectation that we have for this biomarker. Now, when we ask what happens to individuals who were initially called as TMB high from the tumor, but then were recalibrated as TMB low, what does their survival look like? Those individuals are shown here in orange. So they had a raw high TMB estimate and then were kind of corrected to TMB low. And you can see that these individuals have significantly worse survival outcomes than the individuals that remained TMB high, so that did not have false positives in their estimate. And in fact, this orange curve is pretty much no different than the blue true TMB low individuals. Again, this is a cohort of 1,600 patients who all received immune checkpoint inhibitors. And so what this is saying is that for this orange group, they were or may have been recommended for immunotherapy based on a TMB high estimate that was inflated, and that this was actually may not have been the optimal treatment for them because their survival is essentially the same, as the individuals who were TMB low to begin with. And so this bias and inflation in the TMB estimate may indeed have a clinical impact because it's identifying individuals who then don't respond any better than the background group. And so again, just to kind of conclude on this section, we've shown that these tumor-only estimates of tumor mutational burden, or TMB, are upwardly biased. I think this was something that was appreciated in the literature, but we've additionally shown that they can be biased as a consequence of the individual patient's ancestry. And I don't think this was appreciated in the literature, and that this is ancestry that can be inferred from the tumor itself. We developed an algorithm to attempt to recalibrate this estimate, and we show that when you recalibrate individuals back to TMB low, you are able to identify a subpopulation that does indeed have worse survival than they would be expected. Of course, the gold standard is actually doing tumor normal sequencing, but this is potentially a stopgap to identify individuals that have a biased estimate. And so with that, I'm happy to take your questions, and just want to acknowledge the folks that worked on these projects, and as always, the participating patients without whom we couldn't have done any of this. Thanks.
Thank you, Sasha. You're an outstanding communicator. As a communication scientist, I'm saying this, that's great. That's wonderful. Even I can understand what you're saying. So my name is Vishwanath. I'm one of the co-PIs, and I'm supposed to moderate this session. So you can go and say. Just kidding. So questions? Dr. McCarthy.
I know a little bit about this now. I actually know quite a bit. Sure. So, I'm going to ask you, is this on? Yes. The missing data question, because I'm sure you're expecting that. So I noticed in your Kaplan-Meier curves, there was a lot less patient information represented there for the African-American ancestry group. So how do you deal with that? And do you think some of these efforts, like the All of Us thing that NIH is putting on, is going to help with that, or maybe not so much?
Yeah, I think it's a great question. So it's definitely the case that in pretty much all of these curves, let's see if I can get back to, maybe not. In pretty much all of these curves, we're severely underrepresenting non-white patients. And see if I can find that. And in fact, in part, the reason that we had to go to our collaborators at Sloan Kettering is because initially we didn't even have enough data at Dana-Farber alone to just show that this phenomenon is present. I think that the solution is to collect much more genomic data from diverse cohorts. That was definitely the intent of the All of Us, and it is more representative than like this NOMAD reference panel, for example. But it is still a predominantly white cohort, even though the intent was to oversample or ascertain for minority populations. I think part of my view is that having tools that can be applied to a diverse range of data collected in lots of different ways, potentially low quality data that's been collected in places that don't have as much kind of structural support is one way to get around this, basically to just start looking for data that we should have been analyzing that we haven't with new algorithms. But I do think that ultimately these biases get resolved by just investing in more data collection in those populations.
Yeah.
Sasha, very nice work. My question is somewhat related. I'm wondering how you would get at data that maybe comes from an indigenous group or individuals that are actually mixed, you know? So
Yep.
for instance, a lot of Latin Americans are European, indigenous, and potentially also African.
Right.
And so how do you deal with some of that data?
Yeah, so it's a really interesting question. I mean, I think on a technical level, what we worked here, everything I showed was from African American individuals who are primarily 80% African ancestry and 20% European ancestry, and East Asian individuals who are primarily of East Asian ancestry. As you said, Hispanic and Latino populations have a much more complex admixture. I think, again, we have now good algorithms for estimating that genetic ancestry, and we can incorporate continuous values of ancestry for each individual into these approaches. So one thing to do is just to sort of update our algorithms to work with continuous ancestry and admixture. To the first part of your question, which is how do we actually get data, especially from Native American populations, that, you know, I think it's a challenge in part because I think that the field of genetics and in particular population genetics has had sort of, you know, a bunch of screw ups in that area, in that data was initially collected for clinical purposes and then was used for population genetic analyses that it was not approved for. And this had like a decade-long impact on the ability to recruit and work with those populations. You know, I think that first of all, we just have to acknowledge those screw-ups and put systems in place where when we consent people and we explain to them what we're collecting data for, we also assure them that we're only going to be using it for that purpose. I think that actually this is something that the All of Us cohort did a really good job with, which is that before even collecting any data, they did an enormous amount of outreach to Native American populations, and then they made that data publicly available in a sort of staged way as the participants were comfortable with it becoming more available. So I think some of those approaches can be borrowed for other efforts too. I think just fundamentally talking to and engaging the people in these communities in the research itself so they know where everything is going and why, as opposed to sort of, you know, parachuting in, grabbing data, bringing it back to our institutions and working on it. I think that's a really fundamental part as well.
Thank you very much. And to your last point, I'm wondering if you've thought about ways to actually bring this to communities, because I think that the clinical impact that you talk about has clinical relevance for treatment options for particular populations. And I wonder if you thought about how to take this really interesting finding about the clinical relevance of being misdiagnosed for that high group and actually being a low group, how that would impact your choices, what decisions you make for treatment, whether or not there are other treatment options, et cetera.
Yeah, it's a great question. And it is something that, I mean, it's an existential question that I face a lot, because I feel like a lot of times we're developing algorithms for patients at Dana-Farber who are already receiving very, very excellent care. And what we should be thinking about is how to deploy those algorithms in places where that kind of care is not available. We have a collaboration now with the ACR Project Genie, which sequenced a lot of the tumors that I was talking about. And they are trying to expand. That also is a population that's severely underrepresented for minorities. They're trying to expand that, and they have efforts working with historic Black colleges to try to recruit participants and also build up research capacity at those institutions. That's in a very, very early stage. So I can't, you know, I don't really have a sense of whether to what extent that's going to address the data gaps. But that's one effort that I've seen and that I'm particularly excited about. We're working with Melissa Davis on that work. I think we have also tried to partner with industry collaborators, actually, because a lot of times the sort of direct to consumer sequencing and data collection is happening in a more diverse population than here. And again, these algorithms can be applied to essentially any kind of tumor sequencing platform. So we've been working with Tempus in this project, and also with the folks at Caris, and to some extent with Foundation Medicine, to try to broaden this out. Again, it's hard because the industry priorities are somewhat different from the research priorities, but I think that's another opportunity to extend this data to people who wouldn't otherwise have it.
Go ahead.
Thank you for your talk. I just have a question about the 30,000 patients that you discussed in the first part. Thank you. So were these tumors collected randomly from males and females, and do you think sex or age of these patients could induce any variability or play a role in this process?
Yeah, it's a great question. So this is all data that was collected as part of sort of, routine standard of care tumor sequencing here at the Farber. And so it's a mix of it's a subset of the population that we generally see here. It is more shifted towards advanced disease. Typically, you get one of these panels when your treatment isn't working. And so that is going to induce biases in terms of when people enter the cohort, in terms of the stage that they're at, and also just like where in the course of treatment they are. If things are going great, you usually don't end up getting one of these onco panels. So I think that's actually very important whenever we're looking at any of these survival curves, is to think about how the cohort comes in. To your question about sex, so I think I'm not sure if we've really interrogated that directly. I think it's a really interesting question. And, for the technical biases that I was describing, their ancestry is really the key confounder. But certainly this idea of interactions between the environment can be applicable to sex as a kind of surrogate for the environment or for biological differences. And so I think that is a very interesting direction to pursue. Yeah, we haven't done it, but there's certainly, the data is there.
The last question. Is it Sitara?
I think there are a lot of questions on Zoom.
Yeah.
We'll also take, Sitara, go ahead, and then we'll take the question on.
Oh, okay, thank you. Yeah, thank you for the presentation. I'm wondering how the reference populations are determined. Like, you mentioned Japan for Asia, and I'm thinking, Asia is a big place, and
Yeah.
there's East Asia and South Asia and Southeast Asia, and even within those, I'm just wondering how that's determined.
Yeah, no, it's a great question. And Africa also is obviously much bigger than just Yoruba as well. So yeah, I mean, I think it's you kind of actually, you sort of see it even in this, so in this, I don't know if it's obvious, but in this principal component analysis, this kind of cluster over here are actually South Asian individuals who are kind of get projected into a slightly different space. And then in the clinical data, they're just labeled as Asian. And then, a clustering algorithm wouldn't really know necessarily what to do with them. The reference data that we're working with was really ascertained, now almost 20 years ago as part of the 1,000 Genomes Project and the HapMap Consortium. And that was intentionally meant to collect relatively small populations with as much geographic diversity as possible. That doesn't actually, I think in hindsight, there may have been better ways to sample that data to get more interesting genetic diversity. And definitely African populations, South Asian populations, and actually Middle Eastern populations are basically like not represented in those cohorts at all. That is something that I would say has changed quite a lot with genotyping data collected as part of biobanks. So, I brought up the All of Us a bunch, but that's a cohort of 500,000 people. And just incidentally, you're going to have Middle Easterners within that group, and you can construct, and people have constructed reference panels from those populations as well. So I think actually, to some extent, this is fairly out of date. And now that newer data is available, we could get much more fine scale ancestry references. There's also interesting questions about within European ancestry, Ashkenazi Jewish ancestry, for example, as well, that could be asked. I think the data for inferring those components is now available.
Just we'll take one more question at the top. Maybe you can respond to that one.
Yeah, let's see. Sorry, which one? Oh, this. Okay, yeah, I'll read it out. So can you speculate the impact of TMB and ancestry on the design of next gen of cancer vaccines? Is TMB more important than ancestry things? That's a great question. I don't know. I honestly don't know. I mean, I think we so far understand, well, to be honest, what we know about TMB in solid tumors is actually very limited. Like we see this observation that TMB high patients do better. We think it's neoantigens, but there's been a lot of attempts to build computational methods around neoantigen prediction and sort of specific matching to patient HLA types, for example. And those efforts have largely not worked. So I actually think even for solid tumor TMB, we don't exactly know why it happens, and we can probably do a better job of identifying some TMB low patients that still respond well. So then, I guess given how little we know about solid cancers, I think even less is understood about cancer vaccines. I guess I'll just add another kind of area where we've been interested in the potential applications is also in pediatric cancers, which are generally very TMB low, and to what extent these biases factor there. But I think there's just more work to be done.
Let's give him one more round of applause. Great, thank you so much. And I'll let Dr. McCaska introduce the poster session.
I'm Joe McCosta. I'm a PI on the UMass Boston side, one of two. So we have a lot of students who have been supported by different training initiatives at UMass Boston and the Dana-Farber. Many of them are here. Some of them have posters right over there. So I'll encourage you to please go look at those and talk to them about their work. We also have posters for all of the core resources associated with the partnership. And so please go over there and take a look to see what we have to offer to help you with your research. Also, we have our DEI toolkits on the table back there. I lost track of where it was. So please go back there and peruse and take the leaflets and the information that that is important to you. We also have way in the corner some information on Pathfinder. Is there anyone here from Pathfinder? Okay, well, I think it's self-explanatory, so please don't ignore it just because it's over in the corner. And by all means, let's have some lunch. It's going to be behind those doors that people are standing in front of. Just kind of push them out of the way. Go get some lunch, and then please talk amongst yourselves, and please go over to that corner where the posters are. Okay, thank you.